Good morning, everyone. How are you? Thank you, Bruce. So it's a pleasure to be here today, and especially to tag team with Dr. Nash. So I'll do part 2 of student-centered biodiversity and give you the perspective of what it's like to be a teacher and what it's like to work with high school students on very sophisticated science in the high school setting. So I am from Sayville High School. I am a full-time science research teacher, which is actually a thing on Long Island. And my program is the Research in Science and Engineering program. It's been at Sayville for about 10 years now. And the program is designed to encourage students to do authentic science research. They don't necessarily have to have mentors. They don't have to go to competitions. Some of them choose to go to competitions, and many of them just decide to have lots of fun in the laboratory. So these are all students that are doing in-house projects. These are some students that have gone to competition, and a few of them who have even done work here at Brookhaven National Laboratory. Jackie Calamaris did work here, John Faris did work here, and we got to go play at the synchrotron light source here a few years back in the crystallography lab when NSLS-1 was still open, and that is actually our superintendent and the high school principal who came along for the fun. So I was trained by Bruce's team 2 falls ago, right? Not this past autumn, the autumn before. And I was also in the DOE ACTS program here, which is a 3-year summer fellowship for teachers, where I was reintroduced to molecular ecology. When I was in college, PCR had not even been invented. So when I was teaching and had students that want to do genetics work, I actually needed to be retrained and learn all of the new techniques. So it was a nice fit to go and get trained at Cold Spring Harbor Labs to bring students or introduce students to be able to do barcoding. And so it was really interesting. This year I had a bunch of 9th graders who came to me and said, oh yeah, we want to do genetics research. And I said, okay, what do you know about genetics? They're like, well, Mrs. Lutz from the middle school says it's really cool, so we should do it. And I said, great. So I had these 9th grade students, and I said, okay, well, so what do you know then about barcodes? And I literally had a student go into his backpack, and he pulled out a Hershey's chocolate bar and said, well, this is a barcode. And I said, okay, good. This is a good place to start. How are you going to do a barcoding project that's related to biodiversity of Long Island, and what does that really mean? And the answers I got, they were trying to figure out how to do barcoding of chocolate bars. So I said, all right, we're going out. We're going outside. We're going across the street to Brookside County Park, and we're going to go take a walk in the woods. And we started to discuss biodiversity and how, what barcoding really means in terms of biodiversity, and then how they could start to formulate a project. They had to come up with their own ideas, and they had to write a proposal. So this is in September. I'm just meeting all of these students coming in from the middle school, and needed them to really start to think about what their project was going to be. So from the 9th graders' perspective, you realize they're just coming out of the middle school, 8th grade into 9th grade. 9th grade, the high school is a big, scary place, and they all of a sudden have to really think about what this is, and they don't even know what it's like to write a proposal. They have no idea what it takes to write a proposal. So they can maybe write a literature review of something they had to read for English, but certainly not a scientific proposal that's going to Cold Spring Harbor Laboratory. So we went for a walk in the woods, and we looked at all of the different ecosystems that they could look at. So at Brookside County Park, it's a palustrin forested wetland, and so we started to talk about food webs in the aquatic system in the water, the open water system, and then the different zones of that system. And then we started to look as you walk away from the edge of the wetlands, how you start to emerge into uplands, and we started to talk about structure, function, values of all of these ecosystems. And these are all concepts that are way beyond, you know, most 9th and 10th graders. So they're trying to process all this, but they're really excited about being in the woods, except for one girl. She was a little concerned that her sneakers were going to get dirty. So I said, well, Mom will wash them. You're going to get dirty in the woods. So we got back, and they had some ideas. They were really excited about the things they saw. We did a quick little plankton tow. We did a little grab from the substrate, threw some things under the microscope, and they were like, oh my gosh, cool, look at this. What's this? What's this? So they got really excited, and they started to think. And that was what got them, you know, really interested in doing this. They finally understood the connection between the environment and where they were headed with genetics. They still really didn't know a lot about genetics. So these are our teams for this year. We have 3 girls, barcoding limnetic zone invertebrates from a pond at Brookside County Park. Then these 2 girls are doing barcoding the fungus among us at Brookside County Park. And then we have these boys that are doing invertebrates, they're benthic invertebrates in Greens Creek to assess biodiversity and water quality. And they were able to come up with those ideas in the beginning. I just had to make sure that our permits were up to date, so I had to resubmit for New York State DEC. We've had permits ongoing with them for this park because it's literally right across the street from the high school, and we've been doing work there since 1999. And then Suffolk County Department of Parks and Recreation, because it is their property, so we need permission for access. So the proposal writing stage was quite the challenge, because the first thing they had to do was, you know, look at some peer-reviewed literature. Now, these kids are 14 years old, 13, 14. And so they start with Google Scholar, and they come up with this abstract. And they're like, oh my goodness, what did I get myself into? There are a lot of words in there that are very scary to a 9th grader. So, you know, I have to tell them, don't be scared by it. Go home, see if you can make any sense out of an abstract, and then look up all those words, and then come back, and we'll try and make a short little story out of the abstract in the language that you actually can speak. And then in a couple of months, this will be the language that you can speak. So there's a whole process that goes along with that. Well, they started reading, and they, you know, became very efficient at going between peer-reviewed literature and just looking up terms and looking up things just in regular Google. And they were like, okay, this isn't that scary. And then they were like, YouTube is a great thing. So they'd go and look for scientists that would talk about, you know, some of the ideas. And they went to the Barcode Long Island site. And so within a couple of weeks, the fear of all of this big science kind of started to dissipate. And then we could actually be productive, because they were really scared. So we went through the writing proposal. You know, the first 4 drafts were pretty pitiful. And then their writing improved, and they got the difference between how to get that scholarly voice, because 9th graders don't have scholarly voices in their writing, compared to what they would say to their friends. I said, you're not writing this as a conversation to your friends. You need to actually develop this technique. And that is still an ongoing process, will be for the rest of their lives. But they're not so scared of it anymore. And then, so they finally, you know, comes time to submit, and they're super scared. Is it good enough? Is it good enough? And they hit the submit button, and you know, within a week, we got the response back. And so they were accepted, and there were very few comments, but you know, it was a peer review process where scientists did return with some of the comments that they would like them to address. And so they were really excited. So they were literally celebrating in the research room that their proposal was accepted. So that has a big impact on young students. So we went out and did their field collections actually prior to the proposal because we needed to make sure that we had them before cold weather set in. We didn't know that it was still going to be 74 degrees the day before Christmas. But so we were looking for colder weather coming in sooner because now we're at the end of September, early October. So it was very interesting. These are the girls that were doing the plankton toes. And for anybody who does a plankton toe, you know that there's supposed to be a little plastic cylinder on the end to capture your organisms. And so the O-ring broke when we were in the field. And I said, okay, well, you have to problem solve on the spot. Figure out how you're going to make your collections. And they're like, where are the gloves? And so they wrapped the gloves around, and this is how they did their sample collection. I said, that's good. I'd like to see those problem-solving skills in the field. So this is how we collected the samples for those invertebrates. And then the students were very happy. We had very long gloves because it was a little chilly that day and very muddy. They were excited to put waders on. And, you know, getting the benthic invertebrates, that's a sieve bucket, and they're sieving out, and they can see all the things moving around, so they got very excited. And the girls who were doing the fungi collection, there's so many different types of fungi at Brookside County Park that they were really getting excited. And I said, have you ever looked at fungi? They're like, no. We never even knew it was here. So, but it was so interesting. It had so many different shapes and colors, and so they really got interested in fungi. And so we had a lot of fun, especially with the shelf fungus. So then the next part, so they took a full day in-house field trip for this. They pulled out of classes all day. And so we came from out of the field and started to do our sampling, you know, cataloging of samplings. They're like, what do we have to do for that? And I'm like, well, we have to take photographs. They're like, how do we photograph these little things? I said, with a microscope and the computer. So we have this, this nice little microscope that I got from Vernier that actually allows you to take these pictures and it'll go up to 80x. And it just hooks right into the computer. So she's using it right here. Just hooks right into the computer. And you can do measurements. You can do all kinds of additional things with the samples. But the girls who did the invertebrates from the water column versus the boys who did the benthic invertebrates, the girls were getting a lot more really cool things to look at. And the boys, every time the girls would get something, they'd be like, oh my gosh, look at this, look at this. And the boys would get up and they'd go running over and they're like, why doesn't our stuff look as cool as that? So there was a lot of intermingling between the teams. And then the boys started finding some fun stuff. And it was a good time in the lab. The girls who did the fungi, these are just some of the pictures of the different fungi they found. And then these are some of the different catalogs that we had from the invertebrates from Greens Creek. So they did have a good time doing that. And then the laboratory training started. And so again, the only pipette they've ever used is a little plastic pipette. They've never used micropipettes. So there's a lot of training in how to, safety in the lab, and then how to use the equipment. So in my lab, I actually do have a PCR machine, and I do have e-gels, and I have a mini spin. We have a vortex machine. We have a dry bath, and then we have the UV illuminator. And these are all the different pieces of equipment that you need to do this work in the lab. But it's a kind of a big learning curve for them to get all at once. So we waited for the training for about a week, and they came in and they just had to learn how to micropipette with colored water and get the right volumes and actually understand what a microliter is compared to a liter. But it all worked out very well, and the safety was the most important thing, so where to deposit the sharps and making sure that you have the goggles on all the time and gloves. So here they are happily working on their samples in the lab, and we had 20 samples. I think half of them are done. Once they extracted the DNA using the protocols in the Barcode Long Island manual, which is excellent, they had to learn about polymerase chain reaction, what primers are, what they do, and how you have to set the thermocycler settings. So this comes from the Barcode Long Island manual, and then how to look at them on the gels. And so, you know, they do all this work and they're waiting for the moment of truth. And so, you know, this is what you hope for. It's got a nice ladder, and then you can see how your samples came out. And we do get this. It's right here. And then every once in a while you get something that looks like this, and then they're like, oh, what did I do wrong? And so, you know, it's like, well, let's call the experts at Cold Spring Harbor Labs and find out if they can give us some tips on how to improve this method. And they're always fabulous at giving us advice. So now we're on, some of the boys just went out and recollected new samples so that they can run new samples in the next 2 weeks. And then they use the DNA subway, the blue line, which is, when you're doing bioinformatics with high school students, this is a dream come true. I've worked with Mega 6. I have the students working with that. And just to get your sequence results to actually run in Mega 6, the work you have to do ahead of time can take days and days and days to get your data set up so that it's clean enough to run. This is just absolutely beautiful to work with. They're able to produce their phylogenetic trees. They finally get the connection as to why they had to get really good pictures, because some of the pictures they take during the cataloging, I'm like, well, that's too blurry. We can't tell what that is. And we really need to be able to reference it well once we get those barcoding sequences back. So it's all starting to come together for them. But this is actually such a wonderful tool. And then in the end, sharing the results. You know, when we teach them the scientific method, we're always like, well, in the end, you've got to share what you learn with the scientific community, even if the scientific community are your peers. And so this was a student who was part of a team last year, when the first year that Barcode Long Island was running, and she was at Cold Spring Harbor Labs here presenting her research. And what she walked away with was just, she was so excited about science. The scientists from Cold Spring Harbor Labs, they come, they talk to the students, they look at their results, they encourage them, they give them really good feedback. And it was just such a wonderful event. Her parents were able to come. So who enjoyed it more, I'm not sure, the students or the parents. But they all had a really good time, and Cold Spring Harbor does a really nice job. They do a reception for these students. They get these beautiful glass medallions that they have on a lanyard and a nice certificate. And it just really gets students, or keeps them really excited about science, which for high school students, that's our goal. So the benefits is exposure to science as a process. They go to classes, you know, everything is, it's tunnel vision. I'm in a living environment class. I'm in a chemistry class. I'm in an earth science class. And they don't see the interrelatedness between all of those classes. And having an experience like this allows them to really see the interrelatedness of all of the sciences and the math and the technology, which is sometimes hard to do in an actual classroom setting at that level. So science as a process and what it means to be part of that scientific process is really important for students. Understanding the proposal writing phase with peer review and feedback is really, really profound for them. It's a new experience and something that will last with them as they go into college. It will help them be more successful in their post-secondary school years. And then learning the field and laboratory methods, you know, usually students have this vision, oh, I'm going to be a lab scientist, or I'm going to be a field ecologist, but they don't understand that in order to be a really good scientist, you've got to actually be able to do it all. And so that opens their eyes for new career paths and things that they can do. And also that interdisciplinary nature of the science and the technology. Interfacing with scientists from Cold Spring Harbor Labs, for science kids, right? So science is like their contact sport. This is like being able to interface with their favorite sports player, okay? So it's a really exciting moment. And then getting that feedback and the recognition for their hard work provides invaluable confidence, motivation, and it gets them to work harder and just keeps them really excited about science. and it gets their families and their parents excited about science. So thank you very much. It's a pleasure being able to do this tag team with Dr. Nash. I thank Cold Spring Harbor Laboratory. That's a huge undertaking, getting all those teachers with professional development and all these student projects. My students for working so hard and their families for supporting them. Sayville administrators have been great, and thank you for listening to me. Thank you.
We have plenty of time for questions. If you would, go to the aisle mics.
My name is Daniel Carpenter. I've read one of Davenport's papers from about 1900. I have a copy of it. And one of the two organisms you won't find in Cold Spring Harbor now are scallops and eelgrass because of the vast changes in the ecology of Long Island Sound. It'd be great if we could get them back, but I'm not sure how.
Do you want to say something towards that?
Yeah, I know that there are efforts out here to get eelgrass back and save it from being flooded. I'm sure there are people in this room who know way more about it than us. I'm not sure how restorable Cold Spring Harbor is, but it would be great if we could figure out a way to do that, yeah.
Before I was a teacher, actually, I was on the eelgrass committee with the Peconic Estuary program. I actually did the eelgrass restoration projects out in the Peconic and worked with Chris Piccarello and Cornell. And so part of the problem with reestablishing eelgrass beds is you have to have the light transmission has to be correct with the depth, and you also have to have the correct soil microbes or the substrate microbes, which may not be there anymore, and you need the right type of flushing to come through the harbor. And I'm not sure what the conditions are there, but those would be the preliminary things that you need to look at, the amount of nitrogen that's in the substrate, the microbes that are there for the eelgrass to actually be reestablished.
We had a talk on eelgrass yesterday.
Yeah.
Oh, good.
Yeah, which I'm sorry I couldn't see.
Yeah,
so
me too.
It's like you just plugged what I just proposed to NIH. We just asked for some money to try to include microbiomes in this project so we can look at things like the microbes in the environment associated with the organisms that are important, obviously, for their survival, but also are largely unknown. And in association with that, if you're looking at this and you're going, wait, the blue line of DNA Subway takes all the data science away from the kids, what we propose to do is to develop the tools to do the microbiome analysis with the students in interdisciplinary teams. So we're going to have, hopefully, if we can get it, biology teachers working with math and computer science teachers with students from different disciplines, get them into teams, and then use the tools that have been developed by the scientific community to answer questions like, what does eelgrass need, which has been studied, but what's inside of this insect that I found in the Pine Barrens, that kind of thing.
Hi, thank you both so much for coming. I'm very excited that our day in the life of the Carmens River, Greens Creek, Peconic Estuary, Connecticut, and Nissaquag River were able to partner with the DNA Barcode, or Barcode Long Island. Would either one of you be able to speak on some of those projects that they submitted or any positive outcomes from the Day in the Life programs?
Sure.
I have to admit that I'm not aware of what people have done in those programs. I know that there are at least 15 projects that were done in association with yours, and I'd love to highlight them, but I haven't analyzed all the data, and a lot of it is current, right? They're working on it now. But they did collect a lot of samples, and I don't know the data, I have to admit, so I apologize.
No, that's okay.
Yeah.
I think the OSSP student celebration in June, they'll probably be able to highlight that.
Yeah.
Awesome,
yeah.
thanks.
Right, and Melissa, also the 3 teams that you saw are part of the Open Space Stewardship Program, your Day in the Life of the Carmens River, but they're still analyzing, so we don't have data yet. We don't have any results yet, but we will for June.
I was wondering how students are chosen. Do they choose themselves? Is this an elective to do science research, especially at the 9th grade level?
At Sayville High School, it is an elective. When they come in from the 8th grade, they may have been accelerated students or they may not have. The science teachers at the middle school level will introduce to their classes that there is this opportunity. There's a small little application that they have to put in, and then depending on the number of applicants and budget, we always have to wait till budget vote, then I can sit down with the principal and say, how many can we accept? In the past, we've never had to turn anybody away. If I need additional support, I'm hoping that I will get that in the future, because I'm right at the point now where I don't think I can take another student. Yeah, it's really, at Sayville, it's an elective. In other districts, I really can't speak to that, but I know there are other teachers here that might be research teachers. I know at least one. So maybe if those teachers could raise their hand and you could take a look around, there's one back there, there's one over here, and maybe you could have a conversation with them and ask them how it works in their district. Thank you.
I can say that almost all of the teams are part of research programs. So they've chosen to take a research course, and then they've self-selected to be on a team in most cases too. I don't know of any occasions where a teacher said, thou shalt do barcoding or something like that. It might have happened, but I don't know about it. So it's meant to be self-selected. The teachers work with the students to build the teams and then work on the proposals. It's meant to engage them in science, not force them to do science.
Thank you.
Hi, I'm Kim Manzo. I'm with Cornell Cooperative Extension. I work with Chris Pickrell. So I just wanted to say, not a question, just if you want to come find me outside, we have a poster and we can discuss eelgrass and bay scallop restoration.
Great.
Thank you.
I just want to say that the excitement that you created by taking them outdoors was followed with challenges, and that builds the excitement. I'm very happy to hear that. I've been a member of the New York State Outdoor Education Association since 1969. I've worked with Nassau BOCES and taken kids on environmental trips to ComSet. Many of them had never been outdoors, and when we got out in the field, the excitement built, and I thank you for keeping it going.
You're welcome, and hopefully we can keep working with everybody in the community on this stuff.
Yeah, thank you.
Yeah, thanks everyone for coming and listening to us.
Yeah. Thank you. Enjoy the rest of the program.